Good morning, good afternoon, and good evening, everyone, wherever you are. I am Sarah Abul Ella, the AAPS VCU Student Chapter Chair. Thank you all for attending our webinar today. On behalf of the AAPS VCU Student Chapter Executive Board, members of the VCU School of Pharmacy and the College of Engineering, I would like to thank Simulations Plus for accepting our invitation and coming and talking to us today and giving us this honor to host them virtually, especially our speakers for today, John DeBella, Simulations Plus Lancaster Division President, and Maxime Lamberdi, Senior Scientist at Simulations Plus. I would like also to thank the person who made this event possible, Cheryl Ain Igata, Business Development at Simulations Plus. Cheryl, take it away.
You did a very nice job with my name. All right. Thank you very much for that nice introduction. And welcome to the Simulations Plus Gastro Plus discussion for the AAPS student chapter of VCU. My name, as Sarah mentioned, is Cheryl Ann Ayagata, and I'd like to extend a really big shout out to the AAPS student chapter for the invitation to meet today. On the webinar today, as Sarah mentioned, is our division president from Lancaster, California, as well as a senior scientist from Basel, Switzerland, and myself from Boston. Before we get started, please allow me to review a few housekeeping notes and a bit of background about Simulations Plus. Simulations Plus is the leading developer in modeling and simulation software and consulting services. We partner with clients from pharmaceutical, academic, biotech, nonprofit, and government agencies across the globe. Our solutions allow scientists to predict key potential drug dynamics in silico, potentially reducing the number and cost of multi-million-dollar clinical trials, speeding up the time to market of effective new medications. Because Simulations Plus believes in investing in the next generation of modelers, we extend a 95% academic discount to academic institutions around the world. Your research is vital to advancing the science of modeling and simulations. Please do reach out to me directly for more information as to how you may take advantage of this program. Our gift to you following today's presentation is a free 30-day trial of the software. There's no better way learning than to unlock a copy of the software and drive it yourself. We also invite you to join the GastroPlus user group on LinkedIn. With nearly 1,200 members, the GastroPlus user group will provide a forum for discussing best practice, Q&A, FAQs, sharing knowledge of the software functionality, application methodology, and more. This is free access to the world's most advanced PBPK modelers and a jewel of a resource for you to take advantage of. That was my cat. Yes, it is true. Simulations Plus is hiring. If modeling and simulations are of interest to you, why not join the leader in this industry? I strongly encourage you to visit our careers page today. Having said that, it is my great pleasure to introduce today's speakers. First up is John DeBella. John is the president of Simulations Plus, located in Lancaster, California. John did his graduate work in biomedical engineering at Case Western Reserve University in Cleveland, Ohio. John has spent his entire professional career working with Simulations Plus. As you can imagine, the network he has is beyond extensive in this industry. Very soon, John will celebrate his 17-year anniversary. He is one of those rare individuals who has enjoyed great business success while at the same time maintaining his world-class scientific acumen. Following John's presentation is Maxime Lamberdi. Maxime Lamberdi is a senior scientist at Simulations Plus. Before joining Simulations Plus, Maxime received a PharmD from Université Paris Descartes in 2015. In 2014, he received his master's degree in pharmacometrics from the same university. He joined the FDA in 2017 as a postdoc in the Division of Quantitative Method and Modeling within the Office of Generic Drugs. This is where he developed his expertise in PBPK models for locally acting drug products and has published multiple papers on ocular delivery models. Currently, Max is pursuing his PhD at Sorbonne University. Having said that, let's please turn our attention to Simulations Plus President John DiBella. John?
Thank you very much, Cheryl Ann, for the kind words in the introduction. And thank you very much to VCU, Sarah and others for this really nice opportunity today to connect with you guys. Give me a second here to share my screen. And then I will also, please, no giggles in the background, but I'll also share my video. I am connecting to you live from my wife's closet in Los Angeles, California, which has now turned into my makeshift office. I trust that everybody has adjusted well over the course of the last number of months, and you've been able to still realize and appreciate the value of the education that you're getting at VCU. You've still been able to connect with a number of colleagues and other industry and regulatory scientists through virtual meetings like this one. And you're set up for returning to some level of normalcy as we get into 2021. What I would like to do today is just talk a little bit about how we've gotten to the point we're at with regards to the use of modeling and simulation to support both internal research and development activities and, more importantly, regulatory interactions. And my presentation is going to focus on some case studies and use examples for oral product development. When I'm finished, I'll then pass things over to Max, who will continue with the discussions, focusing on a growing area for the use of mechanistic modeling and simulation, that being for drugs which are dosed through non-oral delivery pathways. I'll switch over to full slide mode now and get started. As Cheryl Ann mentioned, I've been with Simulations Plus now for 17 years. And when I first started to tag along with some of our founding scientists like Dr. Michael Bolger and our CEO and founder of the company, Walter Waltos, to meet with prospective clients, we were obviously met with a lot of skepticism. You know, how is it possible that you could try to model all of the different biological, chemical, physical processes that are occurring inside of an animal or in humans through sets of mathematical equations? How could you possibly have any confidence in the predictions that you're generating? When we did identify some potential converters to the gospel of modeling and simulation, they would say, well, we'll dip our toes into these waters and see on a project by project basis how modeling and simulation might help. But we're certainly going to be continuing with all of the different experimental activities we've been working on through the years. So in the beginning, it was how can we apply modeling and simulation to support our R&D projects? Well, as the years went by and there were more and more converts and believers and more publications coming out illustrating how modeling and simulation could be used for a variety of different applications, for a variety of different drug molecules, and the validation and the verification of the approaches were being published. we now have a much more receptive audience when we're engaging with a number of companies today, especially at the management level. The questions tend to focus on how can we utilize and maximize the value that modeling and simulation can provide in my development programs. How can I utilize some of the results to help me prioritize the set of experiments that I may consider running in the future? Or potentially, how can I utilize some of these results to help with the discussions I'm going to have with regulatory agencies? Are there opportunities to potentially waive certain studies based upon modeling and simulation results? So today, very much a lot of buy-in with regards to applying these mechanistic models to help drive drug development programs forward. Where we are with some companies and where all companies are going to be heading is towards implementing these kind of tools as standard operating procedures starting from the earliest stages of drug development. Now there are companies like Novartis, like Roche, like GSK and BMS, who are utilizing with their teams of dedicated modelers these kind of tools once they get a set of compounds from the chemist to begin evaluating in early preclinical development. And they're using those tools to then help with the selection of those compounds which they're going to continue forward with. And so now it's about how do we can change our mindset? How do we change the processes that we've worked with for decades by incorporating more and more modeling and simulation? We're only scratching the floor of this. And the use of modeling and simulation and the need for more and more experienced and trained modelers is only going to grow. I think this is also reflected in this slide here that I use in a number of presentations, just kind of illustrating how that demand for PBPK modelers has increased over the last several years. These are slides that I've typically used when we present to our user groups in different countries. So most recently, we did some virtual sessions for the user base in Japan. And so the dates here are reflective of when I did that presentation. Now, it's been a long time since I've done my own job search. So this is by no means a kind of methodical statistical analysis or an all-encompassing job search. But I went to the website Indeed.com, and this was in November of 2018, and I typed in the search term PBPK modeling. And at that time, and I know it's a little hard to see, there were 42 hits that came up. I repeated this exercise for the Japan user group in November of 2019, same website, same phrase, and this time there were about 20% more job postings 1 year later, about 53 jobs where companies were looking for individuals who had experience with PBPK modeling. In the most recent Japan user group meeting a few weeks ago, did the same search, and now we're at about 25% more job postings today, where companies of all different shapes, sizes, markets are looking for scientists or students who are close to finishing their PhD or master's degree studies who have experience in PBPK modeling. And you can see I just pulled a few of those jobs from the search today, whether it's large pharmaceutical companies, mid-size and small-size biotech companies, companies that are in the non-pharmaceutical space, postdoc research opportunities, and then of course, as Cheryl Ann mentioned earlier, companies like Simulations Plus. So if you take anything away from the presentations that Max and I give today, I do hope it's a real desire to continue to learn about PBPK modeling, because this will only make you all the more desirable as you start your careers. And as I mentioned a little bit earlier, what are some of the drivers for not only more and more companies wanting to adopt and incorporate PBPK modeling, but also the number of companies looking to recruit scientists who have this experience? Well, the first is the way in which peers in the industry, especially, have been publishing on their different case studies and their different applications, and sharing best practices and talking about where PBPK models work well and where there are some opportunities for further improvement. And you can see this is a really nice chart illustrating the number of publications over the years that have cited GastroPlus modeling results for a variety of different use cases. And that number just continues to grow and grow and grow as more companies are onboarding scientists and onboarding software to use to support their R&D activities. So there's certainly a push from industry to adopt more and more modeling and simulation, if for nothing else, to help cut costs and potentially save time. But we've really reached a tipping point when it comes to the regulatory push, led by the FDA, also good support coming from the European Medicines Agency, PMDA in Japan, NMDA in China, and other groups around the globe. In 2019, the first guidance document from the FDA that was focused exclusively on PBPK modeling was published. And this outlines for industry some really nice ways in which you could potentially apply your models to help support regulatory interactions. And they go through and describe in great detail what are the expectations when you start compiling your simulation results into a report, what they expect to see so that you can have a more efficient regulatory review process for your drug applications. And you can see on the right side of this slide, just a small sampling of the number of approved drug products which have been supported by PBPK modeling and simulation. And what's interesting is that, that there's certainly a lot of interest in applying this kind of technology to help support drug-drug interactions, whether metabolism-based or transporter-based. But there are real opportunities today to be able to leverage PBPK modeling to support product development questions, whether it's to help evaluate the impact of co-administration with proton pump inhibitors or other pH modifying agents, whether it's during product development or manufacturing site changes, you're wanting to establish product specifications, or more specifically, particle size specifications. Whether it's to utilize modeling and simulation to evaluate the impact of co-administration with food and using some of those results to help inform drug labeling. So the opportunities to utilize these results for applications beyond just DDIs is vast. And that push continues, as we'll see here in a minute, with some of the new guidance documents that are coming out. But in addition to utilizing PBPK to support innovative or new chemical entity research, new drug applications, what maybe isn't fully appreciated is just how widespread the use of PBPK modeling is for generic drug product development. And what we did is we sent out a survey recently to our list of generic drug companies that are using GastroPlus today. So worldwide, there are approximately 30 drug companies in India, in Brazil, in North America, in Europe, that have GastroPlus and are using it on a day-to-day basis. And what we wanted to do is we had two primary objectives with the survey. One was to get some feedback from our users at these generic drug companies on the functionality that they would like to see added or enhanced as we go into 2021 and beyond, help set some of our software R&D priorities. What we also wanted to do is get some more feedback from them on how they've been using the software to help inform regulatory interactions around abbreviated new drug applications, ANDAs. There's not as much information that is publicly available, or at least easy to get access to, for generic products as there are for new drug products. For new drug products, you can go to the FDA website and you can download the clinical pharmacology reviews, you can download the biopharmaceutic reviews. For generic drug products, there's a few more hoops that you have to jump through in order to get that information. And so we decided to go right to the source. And we were very happy to find out that from the companies that responded, which was around 23 or 24 companies, that there have been worldwide 45 ANDAs, which have been supported by GastroPlus simulations. And what you can see here are the different ways in which they've used the modeling and simulation results in their applications. A lot of them have been using it to help justify wider dissolution acceptance criteria. Some have been using it, the software, to do virtual bioequivalence trial simulations as an alternative approach for the demonstration of bioequivalence. Some have been using it to support major CMC changes. They've been switching some of their production work to different manufacturing sites, or they've been making some process changes, and they've been able to use the software to waive some additional BE studies that have been requested by regulatory bodies. And others have just been using it to help support wider drug product specifications, particle size distributions or tablet hardness and so on. So this is really encouraging, and it's not something that many folks in industry are aware of today, is how we can start using these tools for oral product development issues. But we think that this is going to start to change. And one of the reasons why is because the FDA, just within the last couple of months, have released some new guidance documents where PBPK modeling, or a new phrase that is coming out, which is PBBM, physiologically based biopharmaceutics modeling, now has a key focus. So the Division of Biopharmaceutics in the Office of New Drug Products at the FDA has released a draft guidance on the use of physiologically based pharmacokinetic analyses specifically for biopharmaceutics applications for oral drug product development, manufacturing changes, and controls, where now the focus is applying these PBBM, PBPK models to help support some of the formulation questions and potentially use those results for waiving certain studies. And then just within the last couple of weeks, a new guidance document has come out on the evaluation of gastric pH-dependent drug interactions. And in this guidance document, there are dedicated sections talking about how physiologically based PK or biopharmaceutics models can be used in conjunction with some of the studies that you're performing to help inform clinical study designs. This is exciting. And we're going to only see now more and more adoption of this kind of technology as programs are progressing through clinical development. So the outline for the rest of my talk, we'll start going into the weeds a little bit, talk a little bit about how we structure these biopharmaceutic models or PBPK models, what are going to be some of the important parameters that we're looking to try and define. And then we'll go through some case studies that have been published or presented, talking about the use of mechanistic modeling in the early stages of formulation or biopharmaceutics assessment, going all the way through to post-approval and using the results to support regulatory interactions. So let me just set the stage by making sure that we're all on the same page with regards to our definitions for dissolution, absorption, and bioavailability. And I'm sure now this is review. I know several of the professors well at VCU, some of the very best in the world. I'm sure you're getting this, this education and these points are being driven home, but just for the purposes of this talk today, we swallow something into our GI tract, and within the GI tract, what we're going to be doing is capturing overall the dissolution profile of this oral product. And some of the important parameters or considerations that will factor into calculating the overall dissolution will be things like the ionization of the API material, the solubility, both aqueous and biorelevant solubility, precipitation kinetics, which will be important, especially for basic drugs as they empty from the stomach and transit through the intestine. And then there will be formulation considerations, particle size distributions, shape of particles, any excipients that may be included in the products, and so forth. Once we capture and model the dissolution of the drug along the lumen of the GI tract, then we will start modeling the absorption. And in these mechanistic models, the absorption is going to start taking place at the apical membrane, the membrane that separates the lumen from the enterocytes. And some of the important considerations for modeling the absorption and then ultimately how much enters into the portal vein will be things like the passive permeability of the compound, the permeation that will take place through the cells, the transcellular permeability, and also the paracellular permeability for any absorption taking place between the cells. There will be, of course, considerations for the lipophilicity, so the log-D pH profile will be important. And then once we get into the enterocytes, into the cells, lysosomal trapping considerations will be a factor in terms of how much is then getting into the portal system. The villus blood flows will be important, and especially those changes between fasted and fed conditions. Any interactions with transporters, whether it's at the apical membrane site here or at the basolateral membrane site here. And then, of course, within the cells, metabolism or extraction, whether it's by CYP enzymes, UGT enzymes, or others. And then what we're going to do is start modeling how much of that material is going to cross the basolateral membrane and enter into the portal system, which will then be taken up into the liver. And some of the important considerations when we're modeling the liver exposure will be things like liver extraction or metabolism, interactions with any uptake transporters, both at the basolateral and apical membrane sites for the hepatocytes, and biliary secretion. And anything that is excreted in the bile, we then need to be cognizant or aware of its potential to then be recirculated back into the GI tract, modeling enterohepatic circulation. And when things then pass through the liver and enter into the systemic circulation, this is where we would then start to predict bioavailability and, of course, the full plasma and tissue concentration time profiles. And some of the parameters that are going to be important for capturing the distribution and elimination systemically will be things like plasma protein binding, blood to plasma ratios, which can then be used to help calculate tissue distribution, and then systemic clearance. Are there any extrahepatic routes of elimination that we have to consider in our model? some of the key outputs that are predicted with PBPK models, and sometimes are confused a little bit in literature, are going to be the fraction absorbed, which is going to be the percentage of the dose that was ingested that actually crosses the apical membrane and enters into the gut cells. Separately from that, we would be tracking the fraction that is dosed to the portal vein, which will then be how much of that drug material crosses the basolateral membrane. And if we take the ratio of FDP divided by FA, that will equal FG, or the percentage of the dose escaping gut extraction. And then the bioavailability will be what percentage of that dose is available after it has been absorbed across the gut and after it passes through the liver. Sometimes in publications, you'll see some confusion about this being the absorption, when in reality, it's a reflection of the bioavailability. So we have all of these different parameters, both physical, chemical, and or kind of DMPK or ADME related. And we're also going to have a set of physiological or system parameters as well. And what we wanted to try and do is combine all of the available information that you would be collecting as projects progress, information that we might have from different studies and publications, to then set up a virtual human or animal environment to do your simulations. And so the way in which the models are actually structured are illustrated here. So this is a diagram for the oral absorption model. And when we set up this whole body virtual human or animal environment, think about it as consisting of two components. So the first is going to be what we would call the physiologically based biopharmaceutics model, or the mechanistic absorption model that describes our site of administration, which in this case is going to be the gut. And so you can see here, we would have the GI tract defined through a series of compartments from the stomach going all the way down to the ascending colon, where each of these compartments will have a unique physiological consideration. What are pH values, volumes of fluid, bile salt concentrations, residence times, villi density differences for absorption surface areas, pore sizes for the paracellular routes for absorption, enzyme and transporter expression levels. So we're going to take a lot of different sources of physiological data to help build up the descriptions for each of these compartments that make up the GI tract. And what we're going to be doing here is we're going to be using that physiological environment under fasted or fed conditions with some of the drug properties, some of the formulation properties, to then start predicting what is going to be the local exposure along the GI tract. What's the dissolution, the absorption, any extraction that might be taking place. So this is part one. And then what we'll do is we'll connect the second component, which is going to be our systemic model. And that could be something that we define using compartmental approaches. We have a 1 compartment or 2 compartment or 3 compartment model defined. Or this is where we can start to connect a physiologically based pharmacokinetic model, where we have now a series of additional compartments that are set up that describe different tissues and organs, like the kidneys, the brain, the heart, the muscle, and so on. And what we can do is that once this drug has been absorbed into the bloodstream and passes through the liver, we can start using different approaches to try and predict and simulate what would be the distribution of that drug systemically, both in plasma and to all of these other tissues and organs. Now, in GastroPlus specifically, there's going to be, a streamlined and easy to use way for setting up the system models within the software. you're going to be able to work with a number of different laboratory animals: rat, dog, monkey, mouse, mini pig, or rabbit, or humans. With humans, you can see there are a number of different subgroups, different populations, different health status, and also by age as well. And so this is where you have an opportunity to use tools like GastroPlus to then set up a very specific animal model and translate that into some first-in-human predictions. Or once you're in the clinical environment, using your human model that you've developed, for example, in healthy adults, and be able to easily switch to a different population group to help evaluate the PK in those different groups, to help with dose selection as you prepare for your clinical studies. And don't get tricked by the name GastroPlus. Back 20 plus years ago, when we were working on version 1 of the software, unfortunately, we were not able to predict, as good as we've gotten over the years. At that time, we weren't able to predict that eventually this would be much more than just an oral absorption simulation tool. And today we have these mechanistic absorption, or PBBM models, for all of the major dosing sites around the body. And Max will talk about some of these coming up in his presentation. I just will point out right now, and again, Max will elaborate, that all of these non-oral delivery routes, administration through the eye, inhalation into the lungs, nasal sprays, injections, topical administrations, these have all been developed through funded collaborations that we have with the FDA or with industry partners. Today, we actually have 3 concurrent FDA grants for enhancing and validating these mechanistic absorption models for the eye tissue, for the skin, and for the oral cavity administration route. So the system models are going to be provided and are typically used off the shelf by scientists and industry. The information that is going to be important, that will really allow us to distinguish between different drugs, is going to be these inputs that are outlined here. And I'll briefly go through these. So there's going to be a core set of physicochemical or ADME type properties that we have to describe for our drug molecules. And that would be information like the passive permeability that we measure from in vitro studies, for example, solubilities, both aqueous and biorelevant, ionization constants, lipophilicity constants, plasma protein binding, blood plasma ratio. This is a core set of endpoints that will typically be measured at different stages of R&D. And there's an opportunity to utilize in vitro data when you have it available, or there's been a lot of advancements which have been made over the years to build up machine learning models where you can use chemical structures at an early stage to then predict a lot of these physical chemical properties to inform early PBPK models. So there will be a core set of physical chemical property inputs. Then we start setting up our formulation and dose design. What is the dose levels? What are the dosing intervals if we're doing multiple dose studies? Dosage forms for oral products. Are we administering this as a solution, as a suspension, as a capsule, as a tablet, as a delayed release product, as a modified release product? What sort of drug product specifications have we captured? Do we have particle size distributions? Do we have dissolution data measured under different conditions? So we take whatever formulation data we have and plug that into the model. From a pharmacokinetics perspective, as described earlier, we can either utilize traditional compartmental approaches or PBPK methods, and we can take combinations of in vivo PK from animal studies. We can take in vivo PK from human studies if we have it available. We can also take some of our in vitro data from different assays like hepatocytes or microsomes to help scale up some of the clearance inputs to the models. And then depending on, again, the stages and the applications of our models, what we intend to use them for, we might want to collect some additional data for enzymes or transporters, kinetic parameters, inhibition or induction parameters. We may want to try and incorporate whatever PD or toxicokinetic data we have, any therapeutic or adverse effect information to help build up the model. And so all of this information that you see kind of surrounding the GastroPlus name in the middle here would be information that we would be using as a drug program is progressing. And as we inform the model with more and more information, the different types of outputs we can start to generate will expand. Now, on the surface, you might say, oh, this is actually quite a bit of data that we need for our simulations. And the reality is, and one of the main take-home points we make to industry scientists and companies who are starting to evaluate this kind of technology, is that we're not asking you to generate any more data. I'm sure that at different stages in different departments, somebody is collecting this information. What we want you to do is just make better use of it. Instead of trying to make some decisions about how a formulation is going to perform based upon dissolution data alone, or trying to figure out whether or not a compound is going to have elevated PK if it's co-administered with a compound based upon just an inhibition constant. What we want you to do is just take the data that is being collected by the medicinal chemists, that is being collected by the formulation scientists, that is being collected by the DMPK departments, that is being collected by ClinPharm. And we just want it all integrated within the context of this virtual animal or human environment. If anything, mechanistic modeling and simulation has also helped to break down some of the silos that have been established within companies for a very long time. It forces companies to have these intergroup meetings on a regular basis to make sure that the modelers are maximizing the value from the tools, that they have the data that is being collected to properly parameterize. As mentioned, I'm going to now kind of shift gears and talk a little bit through some case studies focused on oral product development, and then pass things over to Max. But before I do that, I just want to make sure everybody is aware of the different ways in which mechanistic modeling, specifically PBBM or PBPK, is being applied today. There's going to be a number of different applications in discovery PK, really now starting from the point at which you sketch chemical structures. Because of what we've been able to do with regards to machine learning technology and having that be integrated with PBPK, we can start leveraging that marriage between the technologies and use structures at an early stage to do some first screening of compounds based upon predicted absorption or bioavailability. And you can see some of the other activities here as well as we start getting into preclinical development. Here, we can start doing some of the translational work across species and get ready to do some of our first in human projection activities. And we can also start using the technology for pharmaceutical development and identifying any biopharmaceutic risk. We can start seeing what would be some strategic approaches for formulation development using tools like parameter sensitivity analysis, for example. And then as we start having more clinical data captured, we can begin to build up the models and simulate for different study designs where food might be involved. We can start building up mechanistic in vitro and vivo correlations and so on. And then, of course, when we get into the later stages of clinical development, into clinical pharmacology, We can start doing our predictions for different population groups. We can start predicting DDIs and so on. So just keep in the back of your mind now as we transition into some oral product development or pharmaceutical development applications, that there's a lot of use in discovery and clinical pharmacology as well. So first short case study here is to just talk about, how our scientists at the early stages of development utilizing this kind of technology to help with biopharmaceutical risk assessment. So you may have already identified, what are going to be some of the clinical candidates. You've done some of your lead selection activities. You now have some data, maybe early data that has been collected in animals following some IV and or solution dosing. And what you want to do is perform some additional assessment based upon BCS, but you also want to start incorporating some of these mechanistic absorption models to help identify formulation strategies and biopharm risk. And so this table here was one of the very first to outline how a pharmaceutical company, in this case Roche, is utilizing mechanistic modeling as a standard operating procedure in their new development programs. So this publication came out way in 2006. I can't believe it's almost 15 years now. So the specific case study that they highlighted, in addition to that table, was a compound R1315, which was in early stages of preclinical development. They've done a few basic PK studies in rats, and they were getting ready to then start to do some testing in larger animals, dogs. And what they wanted to do was kind of evaluate what would be some formulation strategies. This was a compound that, based upon some of their early solubility screens, had a low solubility. And so they wanted to see, from a simulation perspective, could they improve the absorption or the bioavailability if they were to modify the solubility, formulate it with some excipients, some solubilizers, or whether they could improve the dissolution through particle size changes as well. Essentially, they wanted to evaluate whether or not solubility and or dissolution were rate limiting. And they were kind of surprised from some of the early simulation results that, regardless of the particle sizes that they evaluated, going from 500 nanometers up to 50 microns, or the solubilities that they evaluated here, there really wasn't a big change in the bioavailability. The bioavailability was sitting at 80 to 90%. There was some first pass extraction, but otherwise the absorption was pretty complete, regardless of the particle size or solubilities evaluated. And initially they thought, well, we were going to be making some investments to create some enabled formulations, which are obviously more expensive. And if companies can avoid doing that, then that would certainly save on costs and time. But for the purposes of this case study, what they decided to do was perform some tests in dogs for what they called a best formulation, which was a Cremophore enabled solution formulation, and the worst formulation, which was really just drug in capsule, pure drug substance in a capsule. And they gave those two formulations to dogs, and they wanted to see what the PK looked like. And what we see here are the results on the right for the Cremophore vehicle, and the results on the, on the left for the vehicle solution, and on the right for the drug in capsule. And obviously the drug in capsule had a little bit more variability to the results, but when you calculated statistically the AUCs across these dogs, there were no significant differences. The Cremophore vehicle was bioequivalent to the drug in capsule. And so this was confirmation to Roche and really accelerated their adoption of modeling and simulation mechanistically, because they said, you know, we didn't quite believe those simulation results that were presented on this previous slide. This, from all of the BCS work, from all of the historical or legacy experimental work we would do, this compound seemed to have some severe dissolution limitations. But the software or the simulation said, no, in fact, you're not going to be able to realize a big gain with changes to either solubility or particle sizes. And that was reflected in their dog PK studies. And so when they then got ready for their clinical formulation concepts and preparing for the first in human studies, they were able to go with a product that was not as expensive as what they had originally designed. So this is a really nice example of how you can start using the technology in preclinical development to get some ideas and set some strategies as you're getting ready for first in human dosing. Now, another example here would be when we've done some of our first in human studies and we're now preparing for a bridging study and a formulation switch. How can we utilize modeling and simulation to help us design some in vitro experiments that would allow us to then develop formulations that will give us the performance we expect? So imagine now we're in the early stages of clinical development. And so this was some work that was presented by Sun Pharmaceuticals, where they are working on a BCS class 2 compound. And you can see some of the physical chemical property information that they reported, practically insoluble in water, lipophilic with a relatively high CaCO2 apparent permeability. And what they wanted to do was see if they could start to design some different formulations for improved solubility and oral bioavailability. And they wanted to hopefully identify a biorelevant dissolution method or condition that would help screen formulations as they prepare for this switch. And so what they did is, from their early phase 1 trials, they had PK data that was captured for their kind of drug in capsule first formulations. So these were formulations dosed in healthy human volunteers. This is the average PK that came out from the phase 1 studies. And what they wanted to do was, see, okay, here is my in vivo PK. Here on the right is the dissolution of that capsule formulation in a QC method. Here in orange is one of the early tablet prototypes for this formulation switch in the same QC method. So under the QC conditions, their early prototypes for this formulation switch were showing dissolution that was more rapid than the capsules which were tested in these early phase 1 trials. But what they wanted to do was apply mechanistic modeling and simulation to do some deconvolution activities of this available PK data to identify what was the actual in vivo dissolution over the course of the first 4 to 6 hours. And from that mechanistic deconvolution, they would then use this profile here as a target to help then design their biorelevant or biopredictive dissolution method. So they took this in vivo PK data, built up a GastroPlus model, performed a mechanistic deconvolution to find out what was the actual in vivo release along the lumen of the GI tract, and then used this deconvoluted in vivo release to then help design an in vitro method that would allow for a one-to-one correlation between the in vitro release of their capsule, which was in blue, and what they deconvoluted from the PK data. So they designed this in vitro biorelevant method or experiment that matched the in vitro release of the capsule to what was deconvoluted in vivo. In that same method, they then took their early prototype tablet formulation, which under QC conditions had a very rapid dissolution. And now in this biorelevant media, they had a much, much lower dissolution. There were some interactions with that biorelevant method with some of the excipients that were in this prototype tablet formulation that led to a much, much lower dissolution than the target capsule products. So they then redesigned the prototype in the same, and then used the same dissolution method condition to have an in vitro release that would match the in vitro release of the capsule and also that deconvoluted in vivo release. And then when they did the actual tests, so they took this optimized prototype formulation that was designed from the biorelevant method into a new PK study, into a bridging PK study, they were able to find now that the new prototype product was bioequivalent to the capsule formulations. And that's reflected here in the plot on the right. And then you can see the plot on the left shows the dissolution profiles for both the early capsule and the optimized tablet prototype under the QC methods, and the dissolution rates are much closer than what we saw earlier from the first prototype. So this is a really nice example where a company in phase 1 was able to utilize some of that early formulation data from the PK studies to do a deconvolution, apply those deconvoluted results to help design an in vitro method that would allow them to screen and optimize their next round of prototype formulations. And when they took one of those optimized formulations into another clinical study, they were able to confirm the results that the PK was bioequivalent for the new formulation to the old capsule formulation in that bridging study. Another example as we head a little bit later into clinical development is how we could apply PBPK modeling to understand food effect, and with food effect help guide formulation development. And so when we do want to perform simulations where we consider the impact of food, some of the GI physiology parameters that will be changing include stomach volumes, intestinal volumes in the upper GI, changes in pH, gastric emptying, bile salt concentrations, blood flow and liver blood flow and villus blood flow levels as well. And we also need to account for some of these differences based upon the different meal types. So we know that if we're working, if we're eating a breakfast versus a low fat, low calorie meal versus a high fat, high calorie meal, there will be a different impact on the GI physiology. And those changes in pHs, bile salt concentrations, volumes, and so on will impact then what we predict from a PK perspective. And so all of this information will then be built in and compiled so that we can start doing some of the early formulation assessment work to see how can we potentially optimize or adjust our formulations in order to minimize or eliminate any impact from food. And this is important from a development perspective because we don't want to have any special labeling or dosing instructions for the physicians. If we can have a product approved that shows similar exposure under fasted or fed conditions, it just makes things a lot easier from the physician's perspective, from the patient compliance perspective, and so on. So this is a nice example, a publication that came out several years ago from Novartis, where they talked about setting up a design of experiments type approach for one of their class 2 compounds. So here in this table, you see the inputs that were used to help inform and validate the PBPK model. And then once that model was validated, using it and applying it to predict the impact of food at, in this case here, what we see different dose levels, 200 milligrams and 400 milligrams. And what they saw from those formulations that were tested at the different dose levels is that there was a significant food effect observed. And so what Novartis wanted to do was see if there were ways in which they could potentially redesign the formulations in order to minimize this difference between fed and fasted conditions. And so what they did was perform some 3-dimensional parameter sensitivity analysis, essentially set up some matrices with different combinations of parameters. And here we see, as an example, the fraction absorbed on this z-axis as a function of dose and particle size. And this plot on the left is for the fasted state. This plot on the right is for the fed state. And what they identified was, for this drug molecule, there is in fact a path forward for designing a formulation which would produce similar absorption under fasted or fed conditions. The only issue is you would have to manufacture these particles down to a very, very small size. You would have to have your D50s essentially set to around 100 nanometers. But if you could manufacture with consistency these kind of formulations, then regardless of the dose, you would expect to see similar absorption under both fasted and fed conditions. And there's been a lot of work over the past couple of years that has been published trying to build up more and more confidence with the use of PBPK modeling to support food effect predictions. This is a really nice publication that came out from the GastroPlus user group, authors from 6 or 7 different companies, where they presented a number of different case studies and presented this flow chart, kind of outlining what the strategy should be when you want to apply PBPK models for food effect. The idea here is that industry is trying to push the FDA to think about and consider using more PBPK models to potentially waive unnecessary food effect studies. Their near-term goal is to say, if we can show you a prediction that works well under good assumptions and conditions for fasted and standard meal fed conditions, could we potentially waive some other food effect studies for different meal types? And so I think this is a conversation that is ongoing, and hopefully we'll see some new guidance documents coming out sooner than later from the FDA. I'm just keeping an eye on the clock. I think I have a couple minutes left. Let me just go through this last case study here before I pass things over to Max. And this one now is where we're at the stage of actually having the product approved. But post-approval, we have decided as a company that we are going to be changing our manufacturing process and site. So what we're going to be doing now, we have tested certain batches and lots in different clinical studies. Now that we have the approval, we've identified a more efficient way of scaling up production that includes an inline milling step, which is added to the crystallization process. This new manufacturing step and also the change to different sites results in some new lots that are produced, which have particle size distributions that were different from the lots that were tested in the earlier clinical studies, from the lots that were actually approved. So initially, the FDA said, we're going to have to consider doing another BA/BE study to show that these lots manufactured under these new conditions are bioequivalent to the lots that were tested earlier. Well, the sponsor company, in this case, Janssen, actually put together a modeling and simulation strategy plan. And they said, listen, we've got GastroPlus models developed for this compound. And in all the simulations we've done, we don't see any significant impact from this change. Can we put together a report and set of results that will assess the impact of changes to some of those drug product specifications and predict virtual bioequivalence between the new and old lots? And so the FDA said, okay, we'll approve this plan. Let's see what the results look like. And so the tasks were to, of course, build up the baseline model and validate it with the existing clinical PK data for the older lots. Then do some parameter sensitivity analysis work to evaluate the impact of the changes to some of those critical attributes like particle size, particle shape, the standard deviation of the distribution. And then we would run some virtual bioequivalence trial simulations to show when the new lots would be bioequivalent to the old lots and in what cases they would not be, to help us justify our particle size specifications. So the first step, building up the baseline model, it includes some of the same data we've seen on those earlier case examples. So in this case, this is actually a BCS class 4 drug. So there's potentially solubility and permeability issues. And so using the available ADME PhysChem information combined with some of the pharmacokinetic information that was from the animal studies and also a human IV microdose study, importing some of the available formulation information, in this case, a lot of use of the different particle size data from different lots, having that all be integrated within the context of the virtual human models under both fasted and fed conditions. And so some of those baseline results for these older or non-engineered lots are illustrated here on these plots. So we were able to utilize the same model, making changes only for the differences in dose and formulation parameters, particle size distributions. It's able to capture well the observed PK from all of the clinical studies that were performed. And so this is the first step with any project, really, is to build up and validate your baseline models with the available PK data that you have. Once we have good models in place, the next step was to do then the sensitivity analysis runs like we saw on the previous example for the food effect. In this case here, just showing some of the plots, this is the fraction absorbed on the y-axis as a function of the D50 for particle size on the x-axis. And these simulations were done at different dose levels. And what we were able to show is that with regards to the D50, as long as our D50s were at around 30 microns or less, we would have full exposure. It's only once we got to those D50s, which were greater than 30 to 40 microns, where we would start to see some changes in absorption, which would be accelerated as we increase the dose. So we were able to use this information to help establish what the particle size specifications would be. And there's some additional sensitivity analysis runs which were performed, which were done for things like standard deviation and also for the shape of the particles as well. And so this was all done to help establish specifications for the product, focusing on the mean or D50, the standard deviation, and the shape of the particles. And once that was established, the last step was to then do the virtual bioequivalence trial simulations, where in this case, we can create virtual population groups, taking into account variability and different parameters, both drug specific and formulation specific, along with the physiology, and use that to then first reproduce the observed variability from the clinical studies. And so this is one example output from the population simulator. These pink squares are the individual subject PK points from one of the studies. This solid line in the middle is the mean predicted outcome from the virtual population run. And these dotted lines represent the 90% confidence interval. And so we're able to show that the variability inputs to the population simulator are properly capturing the observed variability from the clinical studies. And then from there, it was a matter of running lots of different virtual bioequivalence trial simulations, comparing different combinations of lots from the new manufacturing approach and the old manufacturing approach at different dose levels. And then calculating the common statistics, the geometric mean ratios for AUC and Cmax, and evaluating whether or not they fell within the 80 to 125 limits. And what the FDA wanted to see was not only confirming here positive bioequivalence or situations where you passed, but also situations where you failed as well, to confirm and to validate those product specifications that were outlined in the earlier sensitivity analysis runs. And so compiling all of these results into a report, there were a couple of rounds of revisions requested by the reviewers, some additional simulations that needed to be run. But in the end, the FDA approved the BABE BioWaiver request. And the sponsor has estimated that by doing this activity, they were able to get to market about 12 months before they would have if they needed to do the full trials. We usually will say, hey, boy, what a victory. We were able to save money from not having to do that clinical study. And from the sponsor's perspective, they actually said, well, yeah, sure, that's important, no question. But we're able to get now this approved medication into patients' hands a lot sooner than we would have going through the traditional routes. And so to summarize, there are lots of opportunities to use biopharmaceutics modeling and PBPK modeling to save resources and R&D. Hopefully you've gotten a small taste from a product development perspective of how this could be used in early preclinical to help better invest resources, to start telling a better story as you're preparing for your clinical studies. And then as we get further into clinical development, using these results to potentially reduce regulatory burden and getting to market faster. Cheryl Ann has also already talked about our company and all of the different divisions that make up the Simulations Plus Corporation, and has also mentioned the GastroPlus LinkedIn group. Just as a reminder, it is free, and you do not have to be today an active user. What we're hoping is that more people join the community, more people get interested in using this, and we build out the user base more and more going forward. My apologies for going a few minutes over. It's standard for me to do so as I get excited to talk about these different topics. But what I'll do now is invite Max to talk a little bit about how the software is being utilized for non-oral delivery routes. Max?
Thank you, John. Thank you very much for the talk. As always, it's very instructive, and it's always scary to go after you. So let's go ahead. First, let me thank you also for the invitation. I want to apologize first because my camera is here, but my screen is here. So I'm not polite. I'm just going to try to look at my slides, but sorry about that. So thank you very much for the invitation. And as John mentioned, like the first part of the talk will be focusing on locally acting drug products. And it seems you had interest mostly for ophthalmic and inhalation route of administrations. So we will cover both. And then later on in the talk also discuss about pharmacodynamic modeling in GastroPlus and how we can do it. And maybe have a small discussion on the benefits of including pharmacodynamics within PBPK compared to traditional PBPK approach that are currently done. So first thing first, locally acting drug products. And what are those products actually? Because it's a name that we hear around, that's floating around, but the definition that was provided by the FDA last year during one of their workshops is, it is a drug where the main site of action is local, and that could be the skin, the mucosal surface of the nose or the lungs, the eye, the GI tract. And the common denominator with all of those drugs is that typically you should not be able to detect them in the bloodstream. And as you can imagine, so blood samples are the easiest samples that you can get from a human being to evaluate the pharmacokinetics of a drug. So when we talk about local acting drug product, you have access to very sparse data and very limited information. So that kind of limits your drug development and in terms of efficacy, but also in terms of safety. The other point is, if for some of them, and we will actually, this is the case for both ocular and pulmonary administration, if the API is detectable in the plasma, can we relate on that concentration to estimate the local concentration? And can we use the plasma concentration as a surrogate for what's really happening where we want the drug, so at the site of action? So those questions are main regulatory questions. In the past, the FDA has relied on clinical endpoint bioequivalence studies that are basically phase 3 studies to estimate and to approve generics for those administration. The result of that is, especially for the more complex formulation, there are no generics on the US market for ophthalmic administration of suspension, for example, inhalation of powders. There are no generics on the US market. So there is a huge necessity to create those generics to reduce the price. And for that, one of the initiatives the FDA is pushing, and I just highlighted the last sentence, is in silico quantitative method and modeling and simulation. And actually in that talk that was given by Office of Generic Drugs 2 months ago, they really emphasize on the benefits of using PBPK model for all of those routes of administration. So go much beyond than ocular and pulmonary. It was also dermal, it was long-acting injectables, it was intramuscular, sub-Q, etc., etc. Name it, and they had a talk on it. So it was very interesting. And I find it fascinating to see that the authorities are actually the one pushing the science to develop those models. And they are thinking ahead of the industry on those questions. The thing is, the FDA's mission is to have safe drugs that have the same efficacy as fast as possible on the US market to have a cheap product that is competitive. And for them, they really see PBPK model as the solution to address the question and to have more generics approved. So that this is really a very strong tool. So as John mentioned, we currently have 3 active collaboration with the FDA on the ocular, dermal, and oral cavity models. But just next year, they are publishing 5 grants on these topics. So it is really a big area of interest, and there's a lot of investment made by the authorities on this. I think at some point we will also see a guidance on how to qualify those models and how PBPK model can actually be used in terms of regulatory packages. So the locally acting PBPK models that we have currently in GastroPlus, we have the ocular, the pulmonary, the oral cavity, the dermal, and of course, the model that John described already, the ACAT for the oral absorption, that can be used for GI, locally acting drug products. One more that is not mentioned on this slide is on new modules for intra-articular administration. So overall, with GastroPlus, and once again, as John mentioned, you can cover basically any route of administration that are currently used in clinical studies and clinical practices. So today, we will just focus on those two, ocular and pulmonary. And let's start with my favorite of all, and I'm totally biased on that, the ocular compartmental absorption and transit model. I'd like to give you a brief history about this model and how it started, because I think it's interesting to see where the push is coming for those approach. So in 2008, the very first version of that model was created by Simulation Plus scientists in collaboration with the big pharma Pfizer. In 2014, Simulation Plus signed their first collaboration with the FDA Office of Generic Drugs through a grant to improve that model and make it more usable for generic drug research in general. With that, we ended up with a better OCAT model, and actually now we are on the third version of that specific model. So this is actually when I joined the development of that model in 2017. After that grant, we had a contract with the FDA to include more mechanisms, such as protein binding. We create new formulations, and we also consider a more biopharmaceutical approach by including the tears pH dynamic that could impact the absorptions, especially for solution drug products. In 2020, we just had a new collaboration for the ocular model that will last until 2023. So that means that at the end, it's going to be a total of 9 years of continuous active collaboration with the Office of Generic Drugs to first create, then improve, and 3, completely qualify this model for regulatory purposes. So the hope from the FDA scientists and myself and everyone at Simulation Plus is that those models can be used by the industry to have a faster development of their product and to actually also create some new generics for the US market. So that's the overall goal. And now let's have a look at the model by itself and how it can be used. So as you're aware, probably the eye is a very complex organ. It is composed of roughly 10 tissues. And you have here on the right-hand side a diagram of the human eye. So the upper layer is the cornea, and then in the anterior segment, you have the aqueous humor, finally the ICB, the vitreous humor, and all of those tissues that are overlapping each other, the retina, the choroid, and then the sclera. And here, on the left-hand side, you have the representation of the mathematical algorithm that describe the eye tissue, the eye organ. So all of the tissues that are represented here on the right, you can find them on the left, and each arrows represent a specific mechanism that can influence the drug transfer from one compartment to another one. So black arrows are simple diffusion of the compound. The red arrows are convection flow that represent the aqueous humor dynamic. And here, those orange arrows are also convection flow that represent the tears dynamic. So what's interesting is not only that model can describe the ocular penetration of a drug and its exposure in all of the tissues, including the tissue that will be your real target, so it could be the retina. It could be the tears compartment if you're just willing to treat an infection, or it could be the ICB if you're willing to develop a drug for glaucoma. In terms of administration of an API to the eye, there are multiple options possible and that have been developed by the industry. The first one that is the most used and probably that everybody done that once in their life is a topical administration. So it is simple. You just put a droplet on the top of your eye, and that droplet is either a solution, a suspension, could be an ointment, could be a cream or gel, but in most cases it would be a solution or a suspension. And the thing is, this is such an inefficient way to give a drug to the eye. It is actually a terrible way to provide an API. Because when you do that, less than 5% is actually reaching the anterior segment. And by anterior segment, we mean everything that is before the lens. So if you're willing to treat a disease that affects the retina, your exposure will be close to 0 in this specific tissue. That means you will need to give the drug maybe every 10 minutes to achieve a significant exposure. So that's not possible clinically. Besides the topical administration, you could use the systemic administration. So you're taking the drug orally, just as John described, it will dissolve, be absorbed, reach the systemic circulation. And once it is in the systemic circulation and in the bloodstream, it will arrive to the ocular tissues that are perfused, such as the ICB or the retina. And then it can diffuse in the other tissues. The thing is with that administration route is the penetration is also very limited because of the retinal barrier. So it's not ideal either. The third approach that is also used is to inject directly into the vitreous tumor, but I don't know about you, I would not really like to have like a syringes getting into my eye. So this is used for mostly monoclonal antibodies because they have a very long half-life. So the administration can be like 1 per quarter only. But for small molecules, because of the patient compliance, it's not optimal. And the fourth route of administration is transscleral, but it is barely not used. So when you look at that picture, you realize that to actually provide the drug to your site of action in the eye, it is very complicated. And you can imagine also how it is to get a sample in human. The best you could do is to get an aqueous humor and maybe a vitreous humor sample for one patient as he's going through surgery. So you're kind of in the blur here because you have no data. You understand that the drug doesn't really go anywhere, but you still want to develop those drugs. And this is where modeling and simulation can become extremely handy because you can run in silico experimentation that can help to improve your formulation before you go preclinical or clinical. And you can also look at both the local and the systemic exposure to really check the balance between the local effect that you're aiming to achieve and the systemic side effect that could appear. So why systemic side effect? The way the OCAT model is implemented in GastroPlus, you have 3 blocks. So you have the ocular compartments, so all of those tissues, cornea, sclera, etc. And those are connected to the systemic circulation. That systemic circulation is itself connected to the oral model or the ACAT that John presented. So the transfer from a drug being absorbed through the GI tract into the ocular compartment is done. When you give the drug as a topical dose through nasolacrimal drainage, and that number is actually slightly overestimated because it should be probably between 98 to 99.9% of the administered dose is actually going either directly into the systemic circulation through the nasal mucosa or first into the stomach, then swallowed and absorbed. The remaining of the dose, and we are talking 0.3 to 0.5% of the dose, actually reach the ocular compartment. So we can define two kinds of absorption: the productive, which is what we want to have our therapeutic effect, and the non-productive, that we want to minimize to the maximum, because some of those drugs has to be administered 3 times a day for the rest of your life. So you could actually have some significant systemic side effect. So it's always a balance and trying to find the right ratio between benefits and risk. The other way to administer the drug, as I mentioned, is to inject it directly into the ocular compartment, and then the drug can move back into the systemic circulation. So you can also look at both systemic and local concentrations. So here you have the interface of the OCAT model in GastroPlus. And as I mentioned before, this model is not just like flying around on its own. It is connected to all of the other capacities of that software. So the systemic circulation here, it can be described either as a simple compartmental model or to a full PBPK model. That means that if, for whatever reason, you know your drug has a potential kidney toxicity or heart toxicity, you can actually follow the heart or the kidney concentration time course following the ocular administration. So to start to imagine the capacities you would have in terms of toxicokinetics, pharmacodynamics, and just overall PK. So this is really a very powerful tool. You're only limited by your imagination in terms of the questions you want to answer. For the species that you can describe directly using the model, we have human, rabbit, and monkey. You can obviously create your own physiologies if you want. So if you want to have specific physiologies for, I don't know, maybe the mouse or another animal, you could do it. You can also create specific physiologies to describe a certain disease state. So for example, a patient that has an affected cornea, you could modify the thickness of the cornea and see how it would impact overall your absorption. And you can also, and we will do that in the next version of the software, differentiate between the New Zealand and the Dutch rabbits. Then, if we were on the platform and you would click on any of those compartments, these are the pop-up window you would see. This is where you would define 2 categories of parameters. The compound-specific parameters that, as the name mentioned, will be different whether you're talking about like tobramycin, dexamethasone, any other drugs. And those compound-specific are, for example, the permeability in each of the eye tissues, the systemic absorption rate from the vascular eye tissues, melanin and other protein binding, such as enzyme and transport, if you have some information that we don't have just yet. The second tab here is the physiology tab. And in most cases, this is the one you would not touch that much unless you want to create a specific physiology that describe either a new species or a disease state. And those parameters define the volume of the compartment, the contact surface between that compartment and the one surrounding, the flow rates, and the melanin content, etc. So now, if we take just a step back in terms of the relevant mechanisms that are integrated in the model, you have everything for absorption and transport, whether it goes from simple diffusion, carrier-mediated transport, or convective flow. You have some metabolism. It can be linear, it can be saturable, based on local concentration of enzyme. You have also the melanin binding that is known to be saturable in some conditions. So you have the choice of picking either a linear or saturable melanin binding. So you have a lot of options to calibrate your model and to define it. For the formulations that we have right now into the model, for topical administration, you have eye drops, so liquid suspension and ointments. For the intravitreal administration, you have some immediate release, could be an injection, same, either a liquid or a suspension, or it could be a control release with an implant. And finally, the last administration pathway is the subconjunctival implants. But this one is not really used also because of patient acceptance of those kind of formulations. So for the capabilities of the program, I will not go through all of them. I think it's really important to remember that, first of all, the OCAT model is not, it is integrated with all of GastroPlus features. So once you have developed your model, you can do a parameter sensitivity analysis, you can do population simulation, you can do virtual B analysis, you can do pharmacodynamic analysis, etc., etc. So you have no limitations, and you can really tackle any questions that may come during your formulation development, or any questions you have regarding toxicity, efficacy, etc. The other aspect that the software does for you, it determines the Cmax, Tmax, and AUC for all of your ocular compartment. So there is actually a button in GastroPlus that you can just click, and automatically all of the PK metrics for all of the ocular tissues will be calculated for you. So that becomes handy when you're trying to compare two sets of parameters or trying to compare two formulations. So to show you actually the power of this model, I'll just give you a brief case studies for a drug, fluorometholone. There's a corticosteroid administered as a suspension in human and also in a preclinical species. The background around that little case study is that if you look at the US market for dexamethasone, which is another corticosteroid, there are currently two formulations on the market, Tobradex and Tobradex ST. Those two formulations have differences in the dose, so 0.1% and 0.005%. And very interestingly, if you give those two drugs to the same patient, the aqueous humor exposure should be the same based on bioequivalence studies. That means that even by dividing the dose by twofold, you're able to reach the same exposure, and that's because they change the viscosity of the formulations. So the aim of this little case study would be to first verify an OCAT model for fluorometholone suspension at a dose of 0.1%. And then to design a new suspension with a dose that is 10 times lower, so 0.01%, but you want to have exactly the same aqueous humor exposure. So to do that, there are three phases. The first phase is the model development. And for that, we had some data in rabbit at a dose of 4×10⁻⁵ molar, and data in cornea aqueous humor. And for the cornea, it was actually divided in two, the cornea epithelium and the cornea stroma. Once the model was verified against those data, we could go to the validation step. And as John mentioned in his last case studies, the goal here is to simply change the formulation characteristics. So switching from solution to suspension, changing the dose and changing the particle size that we had. Actually, we had those information in that study, and run the simulation and see how well we can predict those data. If we can predict them well, then the model will be verified. If we cannot, then we need to go back and better understand what's happening for our drug compound. And once we have our model validated for the 0.1 suspension, then we can go into the fun part, trying to design a new formulation. And you have some constraints, because those constraints would be the one you could see working in industry. You have some safety concern. you want the dose to be reduced by tenfold because, as I mentioned, roughly 99% of the dose is actually reaching a systemic circulation. So if you reduce by tenfold your administered dose, then 9% of what you would administer normally would actually reach the systemic circulation. So you can reduce the risk of systemic side effects by tenfold by simply modifying the dose. You want your particle size to be small, less than 10 micrometers, and same because it is known that larger particle size have some local discomfort and side effects. For the efficacy, obviously, you want to have a similar exposure between the two formulations at 0.01 and 0.1. So typically what we would do here, I would fire up GastroPlus and show you how to do that, but we didn't have time to really do this kind of exercise. So I'll just show you the results. Here you have the final estimation of the concentration in the corneal epithelium, stroma, the overall cornea, and the aqueous humor, following the administration of a solution in rabbit. And overall, the model is doing a pretty good job at capturing the data after we have modified a few parameters. But because we modified some few parameters, we need an external validation data set. And this is what we had with the suspension. So here, keeping all of the parameters the same and just switching from solution to suspension, here are the simulations we were able to obtain. We have a really good description of the local concentration or aqueous humor concentration of fluorometallone following a suspension at 0.1 and 0.4%. And also, we could confirm that the model was also able to predict a particle size differences. So here we have a model that was developed the right way, and we already confirmed that we can predict differences in the dose and differences in the particle size. So we have a model that we actually can use to design a new formulation. And this is what we've done. So the requirements are here just to remind you. And here is our target. So the target is to have a suspension, to have the same exposure as a suspension 0.1% with a particle size of 10.4 micrometers. And if you reduce the dose by tenfold, this is possible if you first reduce your particle size by fivefold, and that is in order to have a faster distribution, and also to increase the drainage rate, so meaning that your formulation will stay longer on the surface of the eye from 1, which is the default value, to 0.4. And if you compare the final AUC and Cmax for the 0.1 formulation, they are almost comparable, like a 1-by-1 comparison versus the 0.1%. So that means by using the OCAT model, an industry could literally develop a new suspension, divide the dose by 10, and go to the authorities saying, our exposure will be the same, our safety will be better. So probably using the 505(b)(2) pathway to have a new formulation that's overall will be beneficial for the patient. And here, I'm just linking the drainage rate parameter that is in link to the formulation viscosity. So there's a nice correlation that was established in 1975 that we are still using for this parameter. So to learn more, I will invite you to either reach out to me. I could share those papers with you, but we have published on that model quite a lot over the last 2 years, and we are still developing the software. So just keep an eye on it, because as new improvement will come. I would also invite you to go to YouTube, where we have the Simulation Plus channel, where you can find a lot of material actually for helping you in your research programs. And especially if you're doing ocular modeling, there is this webinar that was given back in June, and feel free to join it. I'm actually going into more details about the model and the case study. So now, and I see that time is running pretty fast, and I want to give time to questions. So for the pulmonary compartment, I will simply describe the model and don't do the case study. as you're probably aware, the lungs are, can be divided in 5 compartments or 5 segments of the respiratory tract. First, the anterior nasal passage, that is right there, so that's basically your nose. Then the posterior nasal passage, oral cavity, larynx and pharynx, so that's that segment right there. Then you have the trachea, finally the bronchioles, and the respiratory bronchiole. What's important to know here is like those 5 compartments variates a lot in terms of physiology. First of all, the size, the airflow, the concentration of blood capillaries that would enhance the absorption of your API. So depending if your drugs depot in the upper segment or the lower segment, your local concentration will vary, obviously, but more importantly, your systemic absorption will also vary quite a lot. The model structure in all of those segments is the same. And you can divide it in like 3 spaces that are the mucus, the tissue, and the blood. Once the drug is dissolved into the mucus, it can penetrate within the tissue where you can have some lymphatic absorption, some metabolism, or some transporters that would either put it back into the mucus or carry your API into the systemic circulation. The other mechanism that we have in our lungs is the mucus is always going from the deeper segment of our lungs back to our throat to be swallowed. So your drugs can actually move up from one of those compartments to another one through the mucus transit, to the mucociliary transit. One other compartments that we don't have currently, that we have in GastroPlus, but it's not on the slide, we have here a gas compartment for inhalation products, such as any gas that would be inhaled will also connect with the mucus, and the API can go from the gas into the mucus and then be absorbed. What's important to remember, and this is for me one of the beauty of modeling and simulation, is this picture seems very complex, but it's even more complex if we think that everything is happening all together. Modeling and simulation is really a way to understand which parameters are more influential for a specific drugs, specific formulation, or a specific patient. Maybe for some patients, because of their asthma or because of a specific age or condition, the mucociliary transit will have a very significant impact. Maybe for some, it doesn't. That means that maybe for a drug, you could be a potential generic in a certain age group, but when you move to another age group, then your bioequivalence would fail. And those are things you can identify through PBPK modeling, and you can generate hypotheses before you even reach the clinical phase. So going back to some of the examples that John mentioned, there is a gain of money because you're not running trials. There's a gain of time because you will not run trials that are aiming to fail. So integrating those models as soon as possible in your development will really help anyone in the industry in their projects. In terms of structure, you can see the structure is, I don't want to say easier, but a bit simplified compared to the ocular model. You have here 4 of the segments that I've described. And if you click here on that checkbox, the nose will appear. So you have your 5 lungs compartments. All of those are connected to the systemic circulation. And from the extrathoracic, a portion of your dose can be actually swallowed into the stomach and then be absorbed exactly the same way John described it in the first talk. So once again, the systemic circulation can also be described as a full PBPK model. So everything I've discussed around disadvantage for the OCAT also applies for pulmonary, but it also applies for all of the locally acting drug product and PBPK models. For the species, for this specific model, we have the human, the rat, the mouse, and the dogs. And once again, if you click on any of these tissues, and I don't want to repeat myself, but I'm pretty sure you're starting to see a pattern. You will have 4 tabs: the compound tab, where you will enter the permeability, the systemic absorption rate, the mucus cell binding that are specific to your compound, and the physiology tab, where you have the surface area, the mucus layer thickness, etc., etc. And once again, you can use that model to create a specific physiology. So to investigate the behavior of your drugs in the healthy volunteer versus a patient. That means after your phase 1, for example, when you have some human data to really nicely calibrate your model, you can start predicting what will be your local and systemic concentration in patients by changing the physiology and including the disease progression into your modeling and simulation. So that can really become a very nice tool. The main limit of the pulmonary absorption or inhalation drugs right now is how the drug is getting depot and where it is getting depot. So there are some models that have been published that are incorporated in GastroPlus. There are 2 of them, the ICRP66 and the Finlay models, that will create some sub-fraction of the dose. So you have your total dose, and it will divide it in 5 fractions that will arrive into the nose, the extrathoracic, thoracic, bronchiolar, and alveolar compartments. And those fractions, you have 2 options. So either they are calculated by those models, or you can also manually specify them. Some of those can also be measured by using some techniques in clinical studies or preclinical studies. So in the software, if you click on the regional deposition calculation, you have all of those different options that are possible. So whether the drug is inhaled to the mouth, the nose, or if you have a custom application, this is where you would enter your information here. So you can pick the model that you want, and it will provide you the regional lung deposition, and this will be predicted based on the particle size, the standard deviation of those particles, and many other factors that are also compound specific. The output, and in that case, we will have 11.5% of the dose that gets into the extrathoracic, and here 34% that goes into the alveolar interstitial. And for example, in that case, because we know the alveolar interstitial has much more exchange with the systemic circulation, we could expect a higher Cmax and shorter Tmax compared to if we had the opposite, 35% here and 12% here. The other factors that we are integrating, but I won't go into full detail, are the size distribution of the drug carrier. And same, more information available online. For the formulations, we have all of your typical formulations that are currently approved in the US. So the solution, the powder, etc., etc. So in terms of capacities, and I haven't emphasized enough on that, especially for the pulmonary administration, this tool can help you to evaluate the independent contribution of pulmonary and gut absorptions, because it's very easy, like in silico, to perform simulations and to set the absorption in the guts to 0. And everything you would see in the systemic circulation would come from the lungs. So that represents an in silico experimentation that could mimic a charcoal study. And that could be really helpful, especially when we think about local concentrations, because that means you can better correlate what is reaching the systemic circulation with what you have in the lungs. You can evaluate the formulation effect. So basically the same strategy of what I've demonstrated for the OCAT model. And you can include other mechanisms such as lysosomal trapping that affects the absorption, etc., etc. So I will just show you this case study. It is for tobramycin. The model was built based on data from a pulmosphere formulation with a dose of 80 milligrams. And what we had to do here is to optimize the systemic absorption rate to make sure we could describe the profile really well. Once we've done that, by simply changing the formulation and the dose, we could simulate a higher dose, so 300 milligrams, for a different formulation that had a different disposition. And that's a pure predictions. So here, not only the formulation is changing, but also the way the drug intake is done is also changed a lot, and the dose. And you can see that you have a very decent pure prediction of the data. So that probably can help you to understand like the power of those tools and how they can be applied in drug development. Once again, to learn more, there is a very nice workshop that was made by John DiBella and Jim Mullins, our lead scientists for pulmonary administrations. So they are all on our YouTube channel if you're interested to learn more. The model is described in much more detail, and the case studies also explained in details. I see we have roughly 15 more minutes, and I want to give some time for potential questions. So regarding pharmacodynamics, I really like this, this paper, not for what is, and that's rare, but not for what is described into the paper, but because of its title. And I think it is a nice way to kind of look back to the very first slide that John presented. The title is Entering the Era of Computationally Driven Drug Development. And this is exactly where we are right now. We are getting into this era where the drugs, those models are getting accepted. We understand what they can do. And people will change their drug development process in order to generate the best data possible for model development and to have a fast track plan. Here on these slides, that paper actually just copied an initial diagram or presentation that was made by Dr. Jusko from the University of Buffalo in 1995 that demonstrates the whole pharmacokinetics, pharmacodynamic relationship. On the PK side, you have the kinetic of your drug disposition and the distribution in the biophase. And both of those are actually accounted in the PBPK model because the distribution in the biophase could be simply the distribution of your drugs within a tissue or even more within the intracellular space of a tissue. So that distribution in the biophase is accounted by using PBPK model. Then once your drug is where you want it to be, it will impact either through a loss or stimulation of an input or output of a biosignal, whatever it could be, and that will induce your drug response. So that was the scheme for PK/PD that was established in 1995. It's still the same we are using, and it's still doing a great job. Another way to show it, and I'm sure you've seen that before when we talk about PK/PD, you have the PK on the left that summarizes what the drug is doing to the body. And this is the relationship between your drug concentration and time. You have the PD, and the PD is what the drug does to the body. And that's the relationship between effect and concentration. So if you implement that relationship within the PK relationship, then you have your PK/PD model, where now you have the effect versus time. In terms of PD models in GastroPlus, we have all of the basic and standard models that are published out there, whether they're direct or indirect. So I will go to the full definition and explaining each of any of those models. I'm sure you're knowing them, or they're all described in the original publications. But the long story short is a direct model is linking the effect directly to the concentration of your API. So let's say you have an effect related to brain concentration. you could use your simulated brain concentration here and link it to an effect. And then you have different models, linear, log linear, Emax, molecular Emax. So once again, I will not develop much about this slide, but this is the typical response you would see for an effect that has an initial value, which is our baseline value in the body. Because we are dealing with a physical system, it has a plateau. And the relationship on the effect versus the concentration is dependent on the concentration. And where you want to be when you develop your drugs, it is in that sweet spot where you're right before the plateau and right above your EC50, because that really helps you to have your optimal effect with hopefully the minimum side effects. However, what happens sometimes, and that can be seen when you plot your PD effect versus drug concentration, is the appearance of an hysteresis. And that means there is a delay between the local drug concentration and the appearance of the effect. I think that one of the most known example is the erythropoietin that can be administered, and you won't only see the effect 3 weeks later once the drug is completely cleared from the body because it has a very short half-life. And why? This is because first the drug has to diffuse into the bone marrow, then it will start to activate some process, and those processes will actually take 3 weeks in order to create new red blood cells that will be observable into the systemic circulation. So between the time of the effect and the time the drug was there, there is a huge gap, and that creates those hysteresis. In terms of mechanism, so you have some indirect models that could be, and same, I won't describe those. It could be sensitization, tolerance, or any other mechanism, like the pharmacodynamic world is much longer. So here is the link of the indirect model in GastroPlus. Same, because of the time, I'm not gonna go through them. Here you have my favorite pharmacodynamic model because it is so simple at the same time it can do so much, which is the indirect response model, class 1 to 4 that was developed by Dr. Jusko. With that model, either on its own or a combination of that model, you can really answer any pharmacodynamic response. So it is extremely powerful. We have also some other more specific model targeted to bacteria killing and growth. And this specific model defines two populations, one that is sensitive to your drugs and one is resistant. So you will see first a huge decrease of your bacteria population and then a rebound because your resistant population will start to grow and the effect here is close to zero. So this is actually the way to look at the appearance of a new bacteria resistant to antibiotics. We have also the precursor dependent model, and those models are typically used when we talk about effect on neurotransmitter, for example. You give a first drug that activates the liberation of serotonin in the brain. If you give a drug a few hours later, there is no more serotonin to be liberated. So your precursor pool is close to zero, so your response will be zero. And here is the interface. So I won't go into full detail. We have also some more documentation on that. But one of the advantages of PD Plus is you can run the simulation through multiple records, and that helps you to calibrate pretty well those PD models, especially the Emax parameter. And once again, one of the main advantages of combining PPK and PD is to, you can pick your PD compartment and you can choose whether you want your brain, whether you want the ICB for the eye, whether you want the mucus concentration in the pulmonary to drive your pharmacodynamic. And that's such a strong asset compared to traditional PK/PD approach that will use the blood plasma as the plasma concentration time course as a surrogate for the local concentrations. So with that, I would like to thank you very much, and we'll be happy to take questions.
I would like to go first. So this question is for John. So I would like to know how the simulations are validated in GastroPlus.
Okay, that's a good question, and I'll start and then invite Max to also provide some feedback as well. We've spent a lot of time over the years compiling lots of different data sets to use to help first develop the underlying equations and the model algorithms, and then kind of sequestered some additional data sets to use for help validating what it is that we've done. So there's going to be some data which is used for helping to build up and optimize the models, the equations, the parameters. And then once those are in place, using some additional data sets to just kind of verify and validate that those equations are working properly and producing, adequate results. So it's going to be using data both at the beginning stages for development, and then for the later stages, these test sets, so to speak, to help with the validation and verification. Max, do you want to add any additional color?
Actually, and that's maybe my FDA background, but I don't like the word validation for PPK model. I prefer the expression like building trust, because you have so many parameters in terms of PPK models. We have maybe what 600, 700 parameters for some models. Mathematically speaking, you will always find a combination that can describe your data. And PPK is like any tool. If you use it wrongly, you may find a conclusion that will be completely wrong, but it's not because the model tells you it works that it's actually okay. So you have to always be critical. And I actually sometimes question my data more than my model simulations, because I know how my model has been built. Because I trust other simulation has performed. And if I try to run a new simulation and I don't capture the data, I don't want to trash everything directly. So it's really like building trust in your model more than validating it. So obviously, as John described, there's a first phase when we develop GastroPlus, as we are trying to run as many case studies as we find, and that validates for us the platform before we give it to a client. But then once you have it for your own projects, validation is never ending, because you will always discover something. You will discover a new mechanism. You will discover a new transporters. Our founding scientists probably have been working over the last 15 years on midazolam PPK model, and he was still editing it last month. So validating is impossible, but you have to shoot for greatness. Just trust it, go with your guts, and go for it. And defend your model as much as you can, because nobody will trust it. If you go and try to say, yeah, my model is maybe, no, trust it, go, just go ahead.
Okay. I have another question for anyone else, I'm sorry. So in slide 14 for Max, if we don't have these numbers that you mentioned in the model, is there a certain default on the software that you can just pop up and use, or in modeling overall, you have to have these numbers before you start?
Okay, so let me bring up the slide 14 so everybody see what you're thinking. Yes, so so we haven't talked about that in today's workshop. All of the physical parameters, they are coming from literature and they are provided in the model. The other main advantage of the way we do PPK in Simulation Plus is that we also combine QSAR models to predict the compound-specific parameters based on the 2D structure of your compound. So that's an all different phase of validation models, and we have a whole team on that. I'm not going to try to explain you how it works because I have no idea, but I just trust them blindly. The long story short is, very early in your development, even if you don't know much about your compound, you can use those software and this software and those PPK models, because we have those QSAR models that can predict those parameters. So you may not be perfect in terms of prediction, and actually you probably will be imperfect in terms of predictions, because if you're too good, too fast, there is something else that's not working. But it can help you to rank different compounds. So depending on your objectives and your question, once you've imported a structure in GastroPlus, you can start play with it and you can start understanding it. You can start thinking about formulation optimization, et cetera, et cetera.
Thank you. I got a question here. So they are asking in academia, because in academia it's much different than industry. So how do you think in academia we can implement GastroPlus? In our school, we have the pulmonary group, and we have also, like in my group, we were doing genetics. So for genetics, we only had our in vitro dissolution. Do you think we can do population simulation using just the in vitro, or we need to have some in vivo data even from the literature?
John, you want to go ahead?
You've got the OGD experience.
So
and
actually,
then I'll follow up.
So actually, the good thing was when you talk about generics is that you probably have some clinical data available somewhere. And you don't need to, especially in the academia, you can just start to play around with those data, and you would first start with a huge literature research and dig any information you may find, and use that to build your initial model. And then you're entering into the IV-IVC space to first make the deconvolution, so better understand what is the behavior of your drugs within the body, like how fast it dissolves. And you can actually use that as a target to develop your in vitro system around it, and to make your in vitro system as biopredictive as possible. Once you have your in vitro dissolution set, experiment setting that is developed, then you can run your experimentation for the RLD and all of the generics you're aiming to test. And you can start to predict the behavior of those drugs before, without doing a clinical trial, actually, using modeling and simulation. So I actually think in academia and thinking about generics, because your resources are a bit more limited compared to big pharma or biotech companies to do clinical trials, using PPK models can really help you to valorize your project and not just working on a new approach or new dissolution model. You can go a step further and try to see how it could actually influence that and spread the word to the community that you've developed a new method that works in specific condition and that overcomes some issues. I mean, we are currently, we just had a collaboration on the oral cavity, and part of the collaboration is to develop a new super fancy dissolution model with an academic group. So there is a lot of work you can do, and any research you do, if you combine it with PPK, I think you can just broaden the spectrum of conclusions, and you can talk to more people at the end.
So does this mean that you are willing to collaborate with academia if we need your help?
We are talking a lot to academia. Like Chair Lian is actually driving this whole academic program that she mentioned at the beginning. I don't know how many groups we are currently working with, but I think it's more than 40. And that would be the low estimation. I think it's actually much more than that. I mean, GastroPlus is used in all of the major PK centers like University of Buffalo, University of Florida, University of Washington, UCSF. So it is used by many other groups, and not just in the US, but worldwide. So just think about how you could use modeling and simulation to fill some gaps. And I guarantee you there is probably a way GastroPlus can do it.
Okay, thank you so much. Serena, I have a question. Can you go ahead and Serena and ask it?
Yes. Do you hear me?
Yes.
Yes.
Hello, thank you very much for the presentation. I'm actually interested in the PCAT part of Maxime's presentation. So I have questions. First is if there is any chance to have a disease model included, like can I put asthmatic patients or cystic fibrosis patients or like any disease characteristic?
Yes. So the short answer is yes. The longer answer is yes, and it's actually easy because if you know those information and what is the impact of your disease on your physiology, you can create by changing, for example, the mucus thickness or the epithelial thickness to represent your disease physiology. Where it can actually be a bit fancier is you can create multiple physiologies that will represent different stage of the disease. And you actually run simulation across time for the same patient as the disease evolve and you can see what would be the impact on the local concentration as the disease evolve. So that's a bit more complicated to do, but it's possible. So once again, depending on the question, there are many options.
Okay, thank you. And then the second question is, in the model itself, I saw that there is no error going from the bronchular compartment to the alveolar compartment.
That's correct. So actually that's on me because I took the screenshot and the parameter in there is 0. If you switch the parameter to anything is not 0, there will be an arrow here.
Okay.
I need to update the slide.
Okay.
I realized that as I was speaking.
Thank you very much.
I guess I could do that.
Thank you.
Hey, Max, I have a question.
Yes.
Yeah, so I remember you talk about a case that you, I can't remember which drug you used, but the dose is 80 milligram. And after you input that, you got the result, and then you change into another drug. Yeah, this one. And you just predict it would be look like this. So like, are there any requirements to do the prediction for another new drug, or do they need to have similarities, or you can just do whatever you want to another drug?
No, so this is actually a great question. Any project will be drug specific. Because the drug-specific parameters are very influential on the overall prediction. So it is sometimes difficult to draw conclusions from one project to another one, unless the physical chemical properties are fairly close. So that's one thing. One of the limitations I would add to those locally acting drug products is at this stage, because there are not that many case studies, and that goes back to your question, Sarah, about validation. We are still learning, and at this stage, you could do some predictions of the exposure, but we are not right now able to perform those like really nice predictions just based on in vitro information. That is doable, and it has been achieved through multiple case studies for all administration. We're not just there yet for locally acting drug products. So one of the limits is that you need a first set of data to somehow calibrate your model. That could be done through preclinical studies and then extrapolated, but you need a first set of clinical data to really calibrate it. And then once you have a nice model, you can go into the prediction phase for other formulations, other dose, etc.
Okay, thank you.
Thank you, Max and John. This was really enjoyable to walk through this with you, you all. Thank you very much for your time. And if anyone has a request for the slides or follow-up, my email is on the screen, I believe.
Yeah.
And feel free to reach out to me. I'm happy to follow up with you, and we will be in touch with a trial of the software. What better thing to do over the Christmas break, right? All right. Thank you again, Sarah, for hosting us, and this was very much our pleasure.
Thank you, Cheryl and Arlene, John, and Maxime for this nice talk today.
Thank you very much. Have a great day, guys.
Happy holidays.
Happy holidays, everyone. Bye.
Bye-bye.
Bye-bye.
Thank you.
Bye, thank you.